For Mohammed Orfan, first of all, thank you very much for the nice presentation. I have two questions. One question is just, I want to have your opinion. Why the poor countries generate waste food in the same quantity as rich countries like Europe and the United States? You have an idea?
Thank you very much for your very good question. But I have no idea in Europe how they tackle their food wastage. But if you see the graph, maybe they have almost same level.
But Nigeria is twice.
Yes, twice.
Yeah. Okay. Yeah. Okay. Another question is about the quality of the waste. You know that you present some transformation of the some waste like peels, like my question is, if I have a waste containing peels and bread and meat, and how we can do to transform this as quality materials?
Yeah, we can do to supplement animal feed, mostly like the waste bread. We can reuse it for animal feeding or some sort of another type of animal feed we can provide. But for peel, sometimes some people or some countries, they process this, wash it, and make because they contain some lignocellulosic material. So this could be implemented as animal supplementation for animal feed as well as for carbon source for microbial fermentation. So the peels could be utilized for further process.
Okay, thank you.
Thank you.
Thank you very much for your presentation. You know, this topic is a hot topic. Is also important. There are many projects in the European Union or national or international. Now, my question is, can we use this valorized waste for the fly production? Okay? Because it's very important for the alternative protein. Lately, I have come across some papers about this, how to, valorize, how to use this waste for the fly production. So what do you think about that?
Yeah, very good question. But I think in some places, these, just like composting, they are using. In some countries, soldier fly composting, have been used for the production of different kind of flies, and these flies could be as a or mealworm. This also has a potential protein source, and all these mealworms and flies, they can be cultivated on these kinds of food waste.
And one more thing, and also, can we use this, the waste for the production of lactic acid bacteria? Okay?
Yeah, sure. So for this, we need to hydrolyze all these waste material into fermentable sugars. When you got these fermentable sugars, then you can easily cultivate any kind of microorganism. It's very simple.
Thank you.
Thank you.
Maybe I have two questions. Yeah, I will first ask one question to the person who is online. Is that okay?
Okay, yes.
Edilson, I have a short question, if you are still hearing us. Edilson, are you hearing me online?
Yes, I listen you.
Okay, I really appreciated your presentation and the way how you are recycling the byproduct as a meal for your quails, and you are hugely improving the ratio of N3 to N6. That's very, very interesting. The only question I have, when we improve or when we have such higher amounts of omega-3 in the meat, then there can be a huge risk of oxidation. My question, did you incorporate any antioxidants, such as vitamin E, in your feeds? Or do you expect to reevaluate that in the future? Thanks.
Thank you for your question. Yes, the treatment for the total replacement soy meal only used to 2% antioxidants, the VTM.
Now, my question was, did you include any antioxidants, such as vitamin E, in your feeds? Because when you improve the ratio of omega-3 in your meat, it's prone to oxidation. And then how are you dealing with that? Or do you plan to have any test to reduce any oxidation of your meat? This was my question. If you don't have the response now, there is no issue. That's
in the formulation, in the group to antioxidant we use, and the was prepared 2 days, the rate had the premix.
Yeah, the premix contains more vitamins, and they are for the health of the animals.
Only vitamins and minerals, but
Yeah.
no antioxidants.
Yeah, but I suggest you to consider that in your future, because the topic on which you are working is really, really relevant, because at the same time you are considering the sustainability and the quality of the meat, but you are just at this moment, I think, improving the nutritional quality, but keep in mind that's the oxidation of meat that contains high amounts of omega-3. They are like prone to this quality defect. Yeah, but very interesting work, and congratulations again. Yeah, thank you. Now we'll be moving and taking other questions from the audience. Maybe we'll be taking 2 more questions because we are in delay this morning. Yeah.
Okay, thank you, Dr. Gagawa. I have just a question for Irfan. Thank you for your nice presentation. As you know, for waste, we have 2 main challenging concepts, which are first the waste diversity and the seasonal variability. Now, do you have any idea regarding one period in both countries, in our country and in yours, which is Ramadan fasting month? Now, for the circular economy approach, do you have any available data regarding this period and the waste amount in Ramadan fasting period? Thank you.
Thank you very much for your nice question, but unfortunately, I do not have any data regarding Ramadan fasting. Yeah.
Yes. Marilise, did you hear me?
Yes, I do.
You're welcome. You're welcome at the Center of Agri-Food Technology. And I thank you for your collaboration with the applied microbiology laboratory, and now with the center. Thank you for your continued commitment. I have a simple question about polluants in wastewater. Did you use green nanoparticles to remove the pollutant? Did you try to use it? Green nanoparticles?
Yeah, in this particular study, we didn't make use of the nanoparticles, but we have another study that's currently taking place where we are looking at the use of silver nanoparticles in treatment processes. So it is something that is being explored at the moment, but yeah, this particular study that I reported on was just the biological sand filters.
Thank you.
The last question from the audience.
Oui.
Go ahead.
Assalamu alaikum. Bismillah ar-Rahman ar-Rahim. I am Habiba Drisi from Tamarast University and lecturer in microbiology field. My first question is to Professor Arfan about any trials, if you have done any trials to know microbial dynamic in bioplastic production. Are there any trials to know dynamic microbial for bioplastic production? The second question is for collagen. You have mentioned that you can produce collagen from waste, and if this collagen can be used for human health. And one question for Ms. Zahra. You choose only 50 degrees for your experiment. And we know that fungi are not stable at this temperature. Did you any additional information about these details? Thank you very much.
Thank you very much for your question. First, I did not clearly hear what kind of microbial dynamics you want to ask.
Can you repeat, please?
What kind of microbial dynamics you want to ask?
Yes, microbial. Microbial, to know which kind of microorganism.
Microbial diversity.
Yes.
What kind of microbes they have capability to produce?
Yes,
to produce
bioplastic, and if this bioplastic can be used in packaging for fermented food.
Yeah, mostly Bacillus subtilis, Bacillus genus, and some sort of Pseudomonas. They have capability to produce bioplastics. And I think maybe in the world there's some practice this bioplastic could be used for packaging processes. And the second question is you have asked about collagen. Yeah. So I did not mention, I have not worked about collagen production, but this collagen can be extracted from animal waste. Yes. Yeah, this could be used as a polymer for other applications.
Okay. Thank you.
Yeah, thank you.
Thank you for your question. Yes, for the choose for 50 degrees Celsius for scarification, it's not random. There are the old statisticals and characteristics for the different type of the enzyme purified. For the trichoderma and aspergillus, for example, for endoglucanase purified, there are a lot of characteristics and stability effect of the temperature and the stability for different enzymes. And about according to these results, and compared with the enzyme produced with Aspergillus and Trichoderma, the lot of enzyme is stable between 30 degrees Celsius, about 60 and 70 degrees Celsius more than 3 days. And this temperature chooses for the saccharification. The enzyme is stable. More than 3 days, donc the enzyme decreases automatically, the activity decreases in the time. But a lot of enzyme is stable, our production also cause. This is your question? Nice.
Thanks to all. Any last question? Okay, we'll be taking one from here. Yeah, please. Thanks.
Thank you. So my question is addressed for Professor Marlays, if she is online.
Marie-Lise, are you still with us?
She's not?
That she left, yeah, sorry.
Okay, so my second question is addressed for Dr. Azouz. It's about, I would like to understand some points in your interested work. So the first one is about the actual status of the production of fermentable sugar on the industrial scale. If you have any idea of the actual status of the industrial scale, of the sources, please?
Yeah, for example, Civitar produce the electricity and export electricity for the sometimes in the Etatic, just producing in the reducing sugar and converted a lot of waste produced in the industrial residues. Just an example, but there are a lot of, so in the moment, because the waste industry causes the problem for the environment, and the Etatic control assist for to valorization this and to find the solution for this, and automatically a lot of industry, for example, Danone, it's, I think they just the last years started to waste treatment the wastewater, and automatically if treatment any was, there are the same results, the objective to find the sources, the new sources for the energy.
Okay, so until now, what is the sources that we are using?
Yes. It's the biomass, linocellulosics as white straw and white bran.
Okay.
But we can choose another different type of the biomasses, but linocellulosic biomass.
Okay, so another question, please.
We have maybe to stop here.
Okay,
invite you just to continue talking during the coffee break. Thanks to all the speakers of this morning and for your questions as well for here, because we are a little bit in delay. That's much appreciated. We invite you for the coffee break and see you at 11:50, please. Thanks.